And so Joy is a third-year senior from Houma, Louisiana. And as soon as Joy graduates, she is headed to UC San Diego, where she is going to study bioinformatics. And so let's give Joy a round of applause.
Thank you for coming. So my project focuses on examining the evolution of Nalidixic acid resistance in the bacterial Vibrio natriegens with a device called the Morbidostat. So what is antibiotic resistance? Antibiotics are used to kill harmful bacteria, but sometimes these bacteria can develop mutations that help them survive these antibiotic attacks. The bacteria can keep accumulating these mutations. Ultimately, the antibiotic will stop working on them. This will lead to high medical costs, prolonged hospital stays, and increased mortality. Since we can't prevent bacteria from gaining these mutations, one way that we can fight back is to figure out the evolutionary pathway, which is the mutations the bacteria gains to become resistant to the antibiotic. There was a study done in 2011 where a group of scientists put E. coli against 3 antibiotics: chlorphenicol, doxycycline, and trimethoprim. They found that chlorphenicol and doxycycline caused the bacteria to develop diverse mutations. But with trimethoprim, the E. coli populations not only developed the same similar mutations, and also in the same order. They did that with a device called the Morbidostat. It's a continuous culture growing device that basically gives the bacteria the best environment to grow their mutations. How it works is the population is represented as the mole right here. Once the population grows to a certain extent, antibiotic will be added, and that is represented as the hammer here. So the population will die, but the ones with the resistant mutations will keep growing, and that is represented with the helmet. So that keeps growing to a certain extent, and then more antibiotic will be added, and that is represented with the mallet. And this keeps going on and on. So during the summer of 2022, I participated in a summer program called the Summer Science Program at Purdue University in Indiana, where we did something very similar to the paper from before, except for the main differences being we used different bacteria and different antibiotics. Our objective was to stimulate the evolution of antibiotic resistance in a safe controlled culture, and then sequence the culture's genome and analyze the acquired mutations. The bacteria that we used was Vibrio natriegens. It has a really fast doubling time, around under 10 minutes, so it's good for experimentation because you can acquire a lot of mutations in a short amount of time. It is also gram-negative, so it's more resistant to antibiotics. And its metabolic function is similar to E. coli, so it's comparable to the first paper. The antibiotic that we used was Nalidixic acid. It's a clonome that can be used for both gram-positive and gram-negative bacteria. This antibiotic specifically inhibits topoisomerase 4 and DNA gyrase, which are proteins in the DNA replication process. It's also synthetic and used to treat urinary tract infections. So those two proteins I mentioned before are important to this project: topoisomerase 4 and DNA gyrase. DNA gyrase specifically unwinds the DNA, and it's composed of 2 subunits, subunit A and subunit B. And it's represented with this picture here. The gene JAR-A specifically codes for the subunit, the blue part right here, for the A subunits. And you should keep JAR-A in mind. Topoisomerase 4 specifically separates the double chromosomes, and it's composed of part C and part B subunits, and is structured very similarly to DNA gyrase, and that is represented with this picture. It's the blue part. So keep variant parsley in mind. While my team and I were researching our antibiotic and bacteria, we came across this paper. There was a study done in 2003 where a group of scientists put E. coli against nalidixic acid, and it found that E. coli gained resistance mutations in the JAR-A and parsley gene. Since nalidixic acid inhibits the proteins JAR-A and parsley codes for, mutations in these genes could cause bacteria to become more resistant to the antibiotic. E. coli and Vibrio natriegens also have similar metabolic functions, so therefore we hypothesized that we would have seen similar mutations when we put our bacteria against nalidixic acid. This is what our initial setup looked like. We would have 7 vials with 1 being the control, and 3 pumps in the back. First pump was the Vibrio. It was the nutrition for the bacteria to grow. Second pump was the antibiotic, and the third pump was the waste pump. But due to a lot of contaminations and breakages, we had to shorten our experiment from 10 days to 5 days, and from 7 vials to 3 vials. So this is what it actually looked like. So how it worked was you have a vial. Oh shoot. So you have a vial, right? And once its optimal density, which is an indirect way to measure how much bacteria was contained in the vial, was around 0.3, then some of the vial contents would be pumped into the waste, and then 10 milliliters of the Vibrio would be pumped in, and then the nalidixic acid would be pumped in as well. If 2 more generations appeared in a 4-hour period, then 33%, then the nalidixic acid would be increased by 33%. This was our optical density chart for vial 1 growth over 5 days. As you can see, the growth was around 0.3 before it got put into the waste bottle and made to grow again. And this keeps continuing until you see this dip right here. What happened was our bacteria kept growing really rapidly for a while, that we actually ran out of Vibrio, and that was why there was a dip. And as you can see here, the bacteria's growth starts stagnating for whatever reason. This was our optical density chart for vial 2 growth, and it showed similar results. During that time, we decided to take freezer stocks of our bacteria, which is basically freezing the bacteria to be sequenced later on. For vial 2, we took freezer stocks of vial 2 at 9 generations, and vial 1 and vial 2 at 130 generations. The reason we took vial 2 twice was because, according to Real BNC, the software that we use to control and view our media, said that our expected minimum inhibitory concentration, which is the minimum amount of antibiotic needed to kill the bacteria, was the highest for vial 2. It was 168 micrograms per milliliter needed for vial 2 at 90 generations, 126 for vial 1 at 130 generations, and 221 for vial 2 at 130 generations. But that was what the computer said. We decided to test it out in real life with antibiotic strips, this thing. And we found that all our vials reached the maximum of the strips, which was 256. So this was our antibiotic strip. And the blue part around it was when the bacteria started dying. What our petri dish looked like was there was none of this blue part. Like the bacteria kept on growing. So we decided to make antibiotic logs where we can add however much antibiotic we wanted to see what the actual range was. We only tested vial 2 at 130 generations because we were on a time crunch, and it was the most resistant, at least according to our software analysis. We tested 400 micrograms per milliliter of antibiotic against it, 600 and 800, and we found that the range to be 400, and 600, which is way above what the computer said. After that, we sent our models to be sequenced at the Center of Genomics and Bioinformatics at Indiana University to be sequenced through Illumina sequencing. So what is Illumina sequencing? So basically, you have your DNA strand, and it gets fragmented and adapters are added on. So now these fragmented DNA, adapter-added DNA, are then put into a flow cell, where all the chemical processes will happen. So the fragments are attached to the oligonucleotides on the flow cell, and then it bends over and starts replicating itself to create clusters. After you have those clusters, nucleotides will be flushed in with different colors assigned to each type. Once it attaches, it will shine a light of its color, and that machine will take a picture and note down whatever color it was. And this will keep on going for each one added. And now you have millions of billions of these reads, which then the computer would look at and then use the overlaps of the reads to create something, the whole genome that looks something like this. And then you would use the bioinformatics software to compare it to the reference genome to figure out the differences and analyze it. The bioinformatics software that we used was the integrated genome browser. We also used a tool called RScan, which we used to find where the possible mutations were located. This is what the integrated genome browser looked like. The top part was the reference genome. The wild-type genome that we were comparing against for this one was VAL1. So this was the first mutation that we found. If you remember from the paper before, we hypothesized that we would see a mutation in jar A, and we did. So it was a guanine changed to a thymine. This was the mutation. This, if you can see it, it's thymine written over here, and this was the reference genome, but then nucleotides. And it says a guanine was there. So it's guanine changed to thymine, and then the amino acid changed from serine to isoleucine. And the mutations in these strains could have caused resistance to build up in the bacteria. This was our second mutation, the PARC mutation. If you also remember, this is also another mutation we hypothesized we would see. This mutation was only found in VAL1 at 130 generations and VAL2 at 130 generations. And they each contain different nucleotide changes. For VAL1, we saw guanine change to adenine, which caused the amino acid to change from arginine to histidine. For VAL2, we saw cytosine change to thymine, but it was a SAD mutation, so no amino acid was affected. Mutations in this gene could have caused bacteria to become more resistant. So those two genes you've seen before were hypothesized, they were expected. But this one was unexpected. It was the deletion mutation in the TET-R-ACRR gene. This gene codes for two proteins where they regulate efflux pumps. Efflux pumps are used to pump out antibiotics from the bacteria. And mutations in that gene could cause the efflux pumps to become more effective in, pumping out the antibiotic and causing the bacteria to become more resistant. This was found in VAL1 and VAL2 at 130 generations. And each deletion mutation was different. For VAL1, there was a single nucleotide deletion of guanine. This caused a friendship mutation that shifted over the gene, and that eventually led to a premature stop codon. For VAL2, we saw not only one nucleotide deleted, but a lot of nucleotides deleted. So this one-scale deletion event and the one nucleotide deletion could have caused something like a loss of function happen and somehow gain resistance to the bacteria. To summarize, we saw our expected JAR-A and PARC mutations, and we also saw an unexpected mutation in the ACR gene, that should be studied for. By studying these effects, we were able to apply the known resistant pathways of E. coli with Vibrio natriegens, and with that in mind, we could use what we found from this experiment and other related experiments to predict resistant pathways of pathogenic Vibrio species and improve treatment regimens. And these are some fun pictures from the program. This was our initial setup before everything started breaking. Thank you.
Right, we have questions for Dora.
So the unexpected mutation you said caused the loss of function, so like how would that help it survive?
Yeah, that was just a kind of a suggestion. I don't really know.
Oh,
Like something, maybe it could have caused something that helped it, but we're just kind of theorizing.
can you go back to the weird dip that you had in your graph there?
Yeah.
Can you explain what that was again?
Oh yeah. So basically the bacteria started kind of dying a bit because we ran out of the food for it. Yeah.
So where in this graph did you add in the antibiotics? Is that every kind of goes up and down?
Okay, so once it reaches the top, we would pump out the vial contents, and then go back down. And then we would also add in the antibiotic and the food, and it started going back up. If that makes sense. Okay.
So if we did this for like a long enough period of time, instead of it being, it's all right now, it's going, it's like level, so it's still working, right? But if it gets more resistant, would it start being an upward slope because you're not killing off as much as you were before?
Well, it was, since it's like more like, it's like measured until it's 0.3, and then it has to be like pumped out of to the waste bottle, so it wouldn't, you won't see like an upward slope.
Oh, because it goes in place.
Yeah.
So why do you think the vial and antibiotic had different results?
Honestly, I don't really know. Maybe, maybe one was just lucky. Sorry.
So, you said you like had to discard a lot of the products because like the setup ended up like not working. Is what happened like y'all didn't have enough food for like all 7 vials, so you just had to like go down?
No, so the machine that we tested was pretty new, so we didn't really know what we were getting into, and so a lot of stuff like kept breaking. Like there was this one part that like kind of, then yeah, we kind of just had to start over a lot.
So if you were to do this again, what would you do?
We expect, like I would expect to have to start over a lot.
How did you learn about the program that was going on at Purdue? How did you connect with Purdue?
So I searched up summer science programs, and this popped up.
Was it an application process?
It was an application process.
How long did it run for? What was the
It
total?
was 5 weeks.
5 weeks.
Yeah. It was really fun.
So where there's that dip in the graph, there's also a bit to the right, there's less increase as quickly. Was that, did y'all,
Wait, which one? This one?
yes, there.
Yeah, it's the growth just starts slowing down a bit for whatever reason.
And now you were, every time you cycled in, you were, depending on how fast it grew, you were putting in more antibiotics if it was growing faster. I don't know if y'all saw that in the methods, but in the methods, as the bacteria grew faster, they got more antibiotics added to it. And so it's gonna suppress their growth theoretically a little more.
So do you
everything about exponential growth, so most bacterial growth plots are logarithmic. But as far as visualizing it, I think you would get more of the stair step.
So what do you study in college?
Bioinformatics.
Tell them what bioinformatics is.
Basically, it's like biology with computer science put together. It's kind of like a little bit of it is like what you saw here.
What are the white gaps?
So that is for Illumina sequencing, you use the overlaps to create of the fragments to create the whole genome. So these are spaces where the fragments are not coded, and that is what is shown here. These are just spaces of the fragments.
But you see throughout the whole thing, she does get coverage of the whole genome, just not necessarily all in one piece at one time.
Any more questions?
All right, let's give